Okay, let's start. So this is the last lecture. Are you guys sad?
We done it.
You're welcome. What'd you say?
We done it. That's why we
You're welcome, Mirja, to come again and Cell bio.
I already took it.
I know you can still take it again. You can always get an A+++. You can be the tutor. The cell bio tutor. Yeah. Actually, that's what we should have, right? We should have. There's never any tutors for 4.8, 4.9, and those are the classes that really need it. Right? Anywho, so we're just gonna finish our DNA. Huh? I was saying that we should have tutors for these classes. I know. Well, all of you guys who get A's and are going to be here next year and want a tutor, email me and I'll see if we can get something going. Because I know it's really hard because most people who are taking these classes are graduating. They don't stick around very long to tutor. Okay. So let's finish this up. Exam 4 is Thursday, 3 to 5. Make sure you're here at 3, not at 3:30. Okay, I can't stand it when I see people come in a half hour late for a final. It makes me want to cry. The grades on Blackboard should be up to date. So the percentage that you're seeing on there should be your grade as it stands right now. I'm getting lots of people who are asking me, oh, well, now I have to take this or that. And, you know, it's your choice. If you think that even if you get 100, you're still not going to get an A, then don't take it. Do not come to the exam because you're worried I'll be sad. Because I get that every year, like 20 people come take the exam, never studied for it, and waste my time because I have to grade it. So it's much easier for me to put in a 0 and drop it than grade your exam, put in a 60 and drop it. So do not come unless you want to take it. And if you're not going to come, email me and tell me you're not coming so I don't waste my energy and, not energy, really, don't waste the paper. I hate wasting paper. Yeah. So there you go. Any questions? Yes.
Did you already drop 2 quizzes and
I dropped 2 quizzes and 1 recitation because in reality there weren't 13 quizzes or 12. Okay. There was a number missing, right?
Drop 1 more.
No.
Yeah, because didn't you say the pretest you were counting on?
I am. It's droppable.
But then we would have had 13.
Well, it's not a quiz. It's a pretest that I let you drop. So if you bomb the pretest, you can take that instead.
What about if we did good on the pretest thing? Can we add that?
Oh, come on. No negotiation. It's like the story of my life where people negotiating with me. And my kids are like, what if I just eat 2 peas? And they still have ice cream. I said eat 10 peas for the ice cream. How about if I just have a taste of the ice cream and no peas? If it's going to make or break you some way, come see me. Because I don't want, if you're at like a 90.4 and dropping the pretest because you failed it is gonna, I don't know, you come talk to me and we can see what you do. Okay, but yes, for those of you who need 10 points to get to the grade you want to be, I'm sorry, dropping another test is gonna do it anyways. Okay, so far this is what we've done. We went through different kinds of DNA damage. We went through different mechanisms of repair, and the conversation we were just having down here about different things, different types of repair, we were talking about how there's 2 categories. There's reversal, and then there's excision and resynthesis. And when you're studying, it's very helpful to categorize things. And so we were talking about what was it, photolyase and methyltransferase. They fall into the categories of reversal because they're just reversing the damage. They're removing the group that's been added. They're splitting the dimer, whatever. Excision of resynthesis, and resynthesis is exactly what it sounds like. You're excising the damaged DNA, so cutting it out, and resynthesizing it. A polymerase is going to come along and resynthesize it using the current template. That other strand is a template. So you've got your photolysis, your methyltransferases, then we've got NER and BER. What category do they fall into? Right, excision and resynthesis. And we talk about how you can repair errors from replication by post-replication mismatch repair, how you identify the old and new strands of DNA so you know which one should be the template. How was that? Methylation, right? Your older DNA strand is going to be methylated, the newly synthesized one is not. Remember, we learned about that also in regards to activation of DNA replication, but that's also useful in sensing new versus old strands of DNA for DNA repair. We learned about two different mechanisms for a double-strand break repair: non-homologous end joining and homologous recombination. And what category does that fall into? Reversal or excision and resynthesis? I would say excision and let's see. I would say it's not reversal. I would say it's resynthesis. I don't know. Is there excision? There's trimming, right? Excision and resynthesis. And so then we started talking about the DNA damage response. So you know your kinds of damage, you know your ways to repair them, but now you need to recruit those proteins to the site of the damage and have them act in the right place. And that's pretty much the DNA damage response. And then the other main thing that happens with the DNA damage response is it will also halt the cell cycle, because do you want to progress in the cell cycle if you have DNA damage?
Nope.
Okay. That was too long of a pause. And then also induce apoptosis, because if you can't repair your DNA and things are really, really bad, do you want that cell to survive and become a tumor? No. No. Exactly. Remember, tumors arrive usually from one cell. It's a little out of control. So, good. So that's what we're doing. And so then as we're going through the DNA damage response, I want you to start thinking about kind of how that connects to the paper we did last week. I'm telling you, I'm in a time warp. Okay. Life is crazy, but we get through it. Okay. So this is just your general DNA damage response, right? So you need to sense, so there'll be some kind of damage, right? It's exogenous or endogenous. What's exogenous mean? Outside, coming from outside the cell. Endogenous, from within the cell, right? So there could be DNA damage coming from exogenously or endogenously, or it could be replication stress, something causing damage, right? All those things we talked about. And then let's say you got a double-strand break. So that needs to be sensed by something, right? Otherwise, how are you going to recruit the repair proteins to repair it? And then that sensor is going to recruit other proteins. So it'll recruit kinases, in this case ATM and ATR. And the role of those kinases is to do what? Phosphorylate things, right? And so it'll phosphorylate different mediators and effectors. That just means proteins that do things. And then you'll get recruitment of other proteins that are going to do other things, and they're going to all work together to basically affect the cell in some way, either apoptosis, cell cycle arrest, something like that, right? Transcriptional regulation. And they're going to repair the damage. Does that make sense? It actually makes a ton of sense. I didn't really learn about the DNA damage response until I was a postdoc. Maybe someone taught it to me and I just didn't get it. That's possible too. But I didn't really get it until I was a postdoc, and that's because I was doing a project where I had to look up at all these kinases, but it kind of makes a lot of sense. You want to figure out a way to see that there's damage, and then get all the repair proteins so they go, and then stop the cell cycle from progressing until it's repaired. Yes.
So ATM is the one that's like looking for damages?
So ATM and ATR are kinases that are recruited. They're sensors, which we're going to learn specifically. That's like MRN, RPA, those things.
And
Those are things that are going to bind to the actual damage. And then recruit and activate the kinases, ATM or ATR. Yep. Any other questions? Right, so sense the damage, recruit kinases to signal presence of the damage. Kinases phosphorylate and activate other proteins. Phosphorylation creates binding sites for repair proteins, and then you recruit checkpoint proteins to halt the cell cycle. So here are two major sensors that we're going to talk about. RPA and MRN. RPA, again, we keep seeing that protein. That's that single-strand DNA binding protein. And it's going to bind, bind single-strand DNA and detect damage, because a lot of damage will result in long-term single-strand DNA. And then it will recruit ATR. And then MRN, M stands for MRE11, R stands for RAD50, N stands for MBS1. So it's a complex of proteins. MRN binds to double-strand breaks. So that's detection of two different kinds of damage. So there's the other damage that results in single-strand DNA. So from like a stalled replication fork, for example. So remember, sometimes when you have some kind of bulky lesion or some other kind of damage during replication, it's actually going to stall because your DNA polymerase can't get by. And so that's where you'll have a lot of single-strand DNA hanging out, and that RPA is going to coat that single-strand DNA, recruit ATR. For double-strand breaks, it's going to recruit the MRN complex. Those are your sensors, MRN and RPA. And then they both recruit different kinds of kinases. Yeah?
So is RPA just a different kind of single-stranded binding protein, or is it the single-stranded?
So RPA, this is in eukaryotic cells. It's similar to REC-A, which we spoke about a bit in bacteria. Yeah, I'm sure there's more than one kind of single-strand binding protein, but this one comes up a lot. Yeah.
So RPA is for single strand and MRN is for double strand?
Yes. Yeah. So as you're going through this, figure out a way to organize everything. I hope it's kind of laid out in an organized way, but as you're going through it, if you can, this is very conducive to like outline format. Okay, any other questions? Okay, so if you have single-strand DNA that comes from having damage and a stalled replication fork, you're going to have RPA binding to that single-strand DNA. You'll get recruitment of, you'll get recruitment of ATR and some other proteins, and then you're going to get phosphorylation of your clamp, okay? And then you're going to get recruitment of repair proteins and translesion polymerase also. So if you get activation of ATR, there's going to be effects on cell cycle control, because you'll get phosphorylation of another kinase, which is a checkpoint kinase. Remember talking about checkpoints? Right? When checkpoints get triggered, what happens to cell cycle? It stops. So then you'll get ATR phosphorylate check 1, and we'll go a little bit further into that. And so that's going to activate check 1, which is then going to phosphorylate things that stop cell cycle. You're going to also get replication fork stabilization through phosphorylation of RPA, Hol1, some other proteins, and then replication origin control. So when RPA is bound to that single strand DNA, actually I realized after doing that Stump the Teacher that the answer was totally here, but whatever. So I was just giving points away last week. Just giving them away. Buckets full of points. So when you have RPA binding to the single strand DNA at a stalled replication fork, then you're going to get recruitment of proteins such as RAD6 and RAD18, and that's ubiquitination complex. That's going to then monoubiquitinate the clamp, also called PCNA, and then recruit translesion polymerase. What does translesion polymerase do? What does translesion polymerase do? Anything?
It allows for replication of damaged
Right, allows for replication of damaged DNA. It bypasses the damage so that replication can continue. So it kind of makes sense when you have a stalled replication fork, you're going to get coding of that single strand DNA with RPA, which will recruit a complex of proteins that's going to ubiquitinate the clamp and recruit translesion polymerase. And then you can bypass the damage, and that's called translesion synthesis. Okay, so that's damage that results in single-strand DNA or from stalled replication forks. So then you've got your damage that your double-strand breaks. And so that's going to be taken care of by a different pathway. And so here you're going to get recruitment, so you're going to get detection. Remember, you're sensing your damage, and then you're recruiting things. So you're going to sense your damage with this MRN complex and get recruitment of ATM. And so then this should kind of sound a little bit familiar to the paper we did last week. ATM is a kinase. So just remember categories of things. ATM and ATR are both kinases. So ATM phosphorylates. Whenever you have a kinase, you should ask what's it phosphorylating? What does that do? It phosphorylates H2AX, histone variant. Does it sound familiar? Right. And so it phosphorylates the histone variant that's near the double-strand break and helps to recruit repair proteins to the site of the damage, right? So there's a double-strand break, and then MRN is going to bind, and because it's sensing it, and it's recruiting ATM, and that ATM is going to phosphorylate those histone tails nearby. Actually, what should be in here is double-strand break, and then, like, if I can put between this first line and second line, I would draw in replacement of H2 with H2AX, right? Because you need to add that histone variant into those nucleosomes. But, well, it's not perfect. So then ATM is going to phosphorylate the histone tails on H2AX, and that is going to recruit other things. And so then you're going to get MDC1, and which is just another protein.
Because I don't know where it went. I'm trying to. I'm sorry.
No, it's okay. Do you want to come in?
Oh, good.
You might want to learn about DNA damage response. And so then you get recruitment of additional MRN ATM complexes, and this amplifies the signal. It's recruiting more sensors, more kinases, and then more repair proteins, including a ubiquitin ligase, always ubiquitin involved, called RNF8, and that ubiquitin ligase is going to ubiquitinate something. It actually ubiquitinates the histones. And then you get recruitment of additional proteins, such as P53 binding protein and P53, and the BRCA1 tumor suppressor. Okay, what does P53 do? It's a transcription factor. It's a tumor suppressor, right? Okay. So we're going to go a little bit into P53 in a minute. Okay, so ATM, so I'm saying, okay, you get sensing of this damage, recruitment of ATM and ATR. Well, so they're going to control cell cycle through activation of check 1 and check 2 kinases. And so they're just kinases that are going to have a negative effect on cell cycle progression. So one thing they do is they'll inactivate CDKs. Oh my God, are you gonna have to remember CDKs? What are CDKs? Cyclin-dependent kinases. They inactivate CDKs by phosphorylating CDC25. What was
It's a
CDC25?
domain on the CDK.
Huh?
It's a domain on.
No. What's CD25? CDC25. CDC25. Oh, it seems like ages ago.
It wasn't in the lump sum.
Didn't it phosphorylate like the binding site? So
Okay,
Yeah,
so
it was-
CDC25 is involved in somehow regulating
yeah.
that phosphorylation. It's a phosphatase, right? Is it a phosphatase? We1 CDC25? We1 phosphorylates an inhibitory location on the CDK, and CDC25 removes that to activate. And so here it's saying that CHEK1 and CHEK2 phosphorylate CDC25. So it inhibits. It's an inhibitory phosphorylation of CDC25. So let's just draw this out for a minute so it all makes sense to you. Actually, I'll draw in here. I didn't bring a pen, did I? Who has a pen for me? Oh no, here. I'll use a pencil. So it's nice when you can kind of like pull some information from what you've learned before and integrate it all together. And so let's try to do that for a quick second. Okay, so we had, whoa. Okay. So you remember we had CDK, right? And what needs to bind to a CDK to activate it? A cyclin, right? Right. But remember, there was this, that you needed to have this activating phosphorylation, and this inhibitory phosphorylation could also happen. Remember that? Kind of. And then you can have, so the inhibitory phosphorylation was done what? By We1, right? Right? Sorry. And then that activating phosphorylation, which we'll just remember, is by CAK, right? But we're not worried about that one. This inhibitory phosphorylation, whenever it's there, you have no cyclin CDK activity, right? But if you need to activate your cyclin CDK, then CDC, am I right? CDC25 is going to, I made like a little inhibitory thing, but CDC25 is a phosphatase, right? Can I get a witness? Come on, guys. Amen, sister, brother. Okay, CDC25 is going to remove that. And so we're saying here CHEK1 and CHEK2 are kinases that are going to phosphorylate CDC25. So under DNA damage, when ATM and ATR are activated, CHEK1 and CHEK2 are going to phosphorylate CDC25. So it can't remove that phosphate group there. And so you have a cyclin CDK complex that's off, right? So that keeps the CDK off. Does that make sense to people? Is everyone following my logic? Yeah, but I see some people nodding. Give me some more nods.
So there are two phosphate groups, it's inhibited, right?
It depends on where. Remember, there was an activating one in this loop.
But
In this, well, yeah, so let's take this confusion out of it. I was just being complete in my explanation. If you forget about your activating one and just think about your this is all surrounding the removal of the inhibitory phosphate, which would normally be done by CDC25, but it can't be because CDC25 has been phosphorylated by CHEK1 and CHEK2. Yes?
So having checkpoint is the same as having a mutated one, for example. Which one?
Is it the same as having a mutated WEE1? Well, mutated WEE1 would imply that it can't phosphorylate. But here you have a phosphorylate phosphate group that's stuck there. So WEE1 did its activity. It's almost the equivalent of having a mutated inactive CDC25. So you've got your phosphorylation by your kinase and your removal of that phosphorylation by CDC25. And it's that removal of the phosphorylation by CDC25 that can't happen. Okay?
So check 1 and 2 prevents the activation of CDK by phosphorylating.
Yes. Yes. Check 1 and check 2. Here, let me put this back on so we can look at it. Check 1 and check 2 prevent cell cycle by preventing CDK activation by preventing dephosphorylation of that inhibitory phosphate by CDC25. And that's through phosphorylation of CDC25. It's like a puzzle, right? You have to move all those pieces to where they go. So this is one way of activating a checkpoint. And so checkpoint activation often results in apoptosis because if you arrest cell cycle long enough, then you will eventually have apoptosis. Okay, any questions about that? Okay, so in multicellular organisms, unrepaired DNA results in G1 arrest, so cell cycle arrest, or apoptosis, and this is controlled by P53. So P53 is a gene regulatory protein, right? It's a transcription factor. It binds somewhere in some sort of regulatory domain, and it activates transcription of a whole bunch of genes. And it upregulates genes that inhibit cell cycle progression or induce apoptosis. Remember, something that's a tumor suppressor should be doing things that would suppress cell growth, right? Such as inducing apoptosis or cell cycle arrest. So this is just showing, I picked this figure because it's highly mutated in cancer. And it kind of shows some of the places where a lot of mutations happen. Does it show here, or maybe I have another one? Oh no, sorry, it's not here, but I have a figure coming up that shows the location of the mutations. But this does show where things happen. So here you'll have, on these serines is where ATM, ATR, or CHEK2 will phosphorylate. And then here, you can have ubiquitination or acetylation. And there's a nuclear export signal. There should be a nuclear import signal too. What else? A DNA binding domain, right? Because if it's a transcription factor, it should hopefully bind to the DNA, right? It also acts as a repressor, not only an activator, because it represses expression of genes that are inhibitors of apoptosis. Again, makes sense. You don't want to have inhibition of apoptosis when you're trying to induce apoptosis. And so it's controlled by post-translational modification, right? So you've got these different functional domains that are regulating the stability and activity of P53. So some things I pointed out. So MDM2 is a ubiquitin ligase, and it will ubiquitinate P53 on these lysines. And so it's going to keep P53 levels really low in the absence of DNA damage. You don't want P53 going and activating all these genes that are involved in apoptosis and cell cycle arrest unless you need it, right? And so P53 is always being made, but it's always being ubiquitinated and degraded by MDM2. That seems inefficient, but it's a good sensor, right? And so you want to be able to, as soon as you need it, boom, stop degradation, and then you have plenty of P53 to activate gene expression. So if there is DNA damage, then you get inhibition of MDM2 activity, usually through phosphorylation of P53, so it can't interact with the ubiquitin ligase anymore. P300 is a hat, and it binds to P53, and it acetylates histones and promotes gene expression. And it also acetylates P53 on lysines, these same exact lysines, and competes with ubiquitin. So again, another way to stabilize. If ubiquitination sites are not available because they're acetylated, you're going to have more stable P53. Okay. So if you have DNA damage, then you're going to get stabilization of P53. So normally, this picture is small, I'm sorry, hopefully you guys can see it at home a little bit better. So normally, MDM2 is binding to P53 and constantly ubiquitinating, it's being degraded, it can't do anything. But when you have damaged DNA and you get activation of all these kinases we've been talking about, then you are going to get disruption of MDM2 and P53, and then you're going to get P53 phosphorylation, stabilization, and then it can go into the nucleus, bind to the DNA, recruit a hat, which will acetylate histones, and that will also promote gene expression. So not only is an activator, it also recruits hat. It also represses other genes, all with the goal of cell cycle arrest and apoptosis. So just to kind of tie this back into cell cycle, remember DNA damage is easily detected at G1 and G2M. We've talked about that in the context of cell cycle, but now remember we're learning about DNA damage. That's when a lot of that is being detected, and then you get that cell cycle arrest. Remember this slide, right? That's your cell cycle again. DNA damage is going to inhibit progression to S and M phase. So all of these places, you're going to get cell cycle arrest if there's DNA damage. That's when this sensing is happening. Why did I put this here again? I must have, sorry guys. Okay. So this is just to go through this again. So we talked about sensing the damage and check 1 and check 2. This is another way to look at it. So if you have a double strand break, remember, or some damage, you'll get ATM and ATR kinase activation because remember it's silenced by RPA or MRN depending on the type of damage. ATM and ATR are going to get recruited and activated. They're going to activate check 1, check 2. And then you're going to get phosphorylation of P53. And then P53 is going to be active and stable. And it's going to go into the nucleus. And it's going to bind and activate transcription of different genes. One of those genes that's going to be activated, we learned this in cell cycle, I'm just trying to bring it back, is P21. Remember? Remember P21? CKI? Right? So cyclin kinase inhibitor. So then you're going to have cell cycle arrest. If your cyclin kinase is inhibited, you cannot progress forward. So try to connect the DNA damage with the P53 with cell cycle. Okay. Is this coming together for you guys? Is it making sense? Or is, yeah, I see nodding. I like nodding. Any questions about this?
Do you remember who removed
Who? Okay. What's your negative regulator of P21? SCF. Yep. Ubiquitin ligase. Yep. Because it gets phosphorylated, recognized by SCF. Yep. Yeah. What stops phosphorylation of P53? Inactivation of check 1 and check 2. So just like things need to be turned on, things need to be turned off. So every time you have phosphorylation and activation of something, there's going to be a phosphatase that's going to dephosphorylate and inactivate. Yep. Good question. Yep, you can't have these going on forever. Okay, how are we doing? Okay, so this was just a close-up of what I just went through for those who have bad eyes. Remember, P53 is a transcription factor. It's normally degraded by MGM2. Phosphorylation prevents MGM2 interaction, so it can't be degraded, so it's stable and can go on and activate gene expression, right? And so this is what we just went through. The outcome of production of a CKI is cell cycle arrest. Check, and this is the other thing I just went through. Check 1 and check 2 also phosphorylate CDC25, and that inhibits its function. So this is what I drew out, right? So normally you have your cyclin, CDK, it's inactive, and then you've got your CAK and your WEE1, but your CDC25 needs to remove that inhibitory phosphate. But if you have phosphorylation of CDC25, wait a minute, right? If you isn't that funny? I'm giving you a picture that shows activation of CDC25 by phosphorylation. But in reality, it's not a perfect picture to explain this, is it? Because then there's, remember, with phosphorylation, you can have activation or inhibition depending on where you're phosphorylating. So sorry, I gave you guys an inaccurate picture. But in this case, in the case of phosphorylation by check 1 and check 2, you're phosphorylating to inhibit. So don't look at this and think it's activating, okay? It's actually an inhibitory. There is also phosphorylation that activates CDC25. That also happens, that's by MCDK. That's what we learned in cell cycle. But then check 1 and check 2, in the case of DNA damage, will come in and phosphorylate CDC25 elsewhere and inhibit its function. Okay. So you can think of ATM, ATR, check 1, check 2, and P53 as master regulators of the DNA damage response. So here's check 1, check 2. These are showing you different mutations that occur in cancer. So remember, when you have defects in your DNA damage response, that's very much going to promote cancer, right? Then we talk about that mutator phenotype. If you cannot repair your DNA, then you're going to accumulate mutations. And so in endometrial, gastric, colorectal cancers, you see these two different mutations in check 1. Check 2 gets tons and tons of mutations all over the place. And it's kinase domain. If you have mutations in your kinase domain, you're not going to be an active kinase. You can't activate. So actually think about it for a minute. If I give you a question on an exam that says, you know, in AML, the kinase domain in check 2 is mutated, what is the outcome? What would you guys say? It won't be able to reverse cell cycle. You have to ask yourself, well, what does check 2 do? It phosphorylates CDC25, right? And so if you can't phosphorylate CDC25, then you're going to have that inhibitory phosphate group stuck bound to that cyclin CDK complex, and then it'll be inhibited, right? But if you can't phosphorylate, then you can't, basically you can't inhibit cell cycle, right? And so then you'll keep replicating despite having DNA damage. So there's no activation of your DNA damage response. You guys are as tired as me. Okay. So that's what I told you. What? It's almost like I added slides twice. So here's some mutations in P53. You can see there's tons and tons of mutations. Each of these different lines right here is indicative of one kind of mutation. And then you can see each region of the protein has a different job. The transactivation domain, SH3 binding domain means it binds to phosphorylated things. interacts with DNA repair proteins, interacts with transcription factors, actually has endonuclease activity. And so if you have mutations in any of these regions that are involved in activation of gene expression, that are involved in interaction with HAT, here's P300, involved in DNA binding, they're all going to inhibit the function of P53. And what happens if you inhibit P53? What's the outcome? Huh?
Colon cancer?
Some kind of cancer, tons of kinds of cancer. Yeah, because remember P53 activates gene expression of all these different genes that are involved in apoptosis, cell cycle arrest, such as P21 was one example. Right? Any questions about this stuff? We're nearing the end. The end is near. 100 million. It's going to be really hard to have a lot of questions. I'm going to try my hardest to give you 25. You guys have until tomorrow, end of day, to send me questions, by the way. All multiple choice, yeah. And a million points in extra credit questions. Oh yeah, you guys are going to rock this. 2 lectures, come on guys. This is the worldwide distribution of P53 mutations in cancer. So you can see there's a ton of P53 mutations in a ton of different cancers. So it makes sense, it's a tumor suppressor. So if it's mutated, it can't work, it can't activate DNA damage response, and you're going to get accumulation of mutations. And that is it. We can use the remaining time